Welcome everybody to another Friends from the Field webinar. Thank you for joining us today. This webinar series is co-hosted by Blue Hill Heritage Trust and Island Heritage Trust. We have been doing these webinars since the end of April. If you would like to see any of the past webinars, you can tune into the Blue Hill Heritage Trust YouTube channel. If you would like to introduce yourself in the chat box, please feel free to do so. You can let us know your name, where you're tuning in from, and why you're interested in today's topic. My name is Jake McCarty from Island Heritage Trust, and today I'm joined by Blue Hill Heritage Trust's intern, Miranda Flora.
Hi everyone, I'm Miranda. I would like to point out a couple technological features for today's webinar. You can see in the bottom of your window that there is a chat option. So if at any point during Rocky's presentation you have a question, feel free to write it in the chat and Rocky will take a moment during his presentation to answer it. In addition, at the end of the presentation, we're going to have 15 minutes or so for a Q&A session. And if you would prefer at that time, you may click on the raise hand feature at the bottom of your window and ask the question to Rocky directly.
Thanks, Miranda. So I think without further ado, I'll introduce our wonderful presenter today, Dr. Rockwell Parker, Assistant Professor in the Biology Department at James Madison University, a primarily undergraduate institution of Virginia. Rocky identifies as cisgender gay man, and he does a lot of advocacy for underrepresented students in his community. He is a chemical ecologist who studies reproduction and physiology. And Rocky, we're so happy to have you here today.
All right, thanks, Jake. I appreciate it. And thank you for the opportunity to speak. I'm happy that everyone wanted to tune in and hear about snake sex and all of the secrets therein. So before I'll start, I just want to say that one of the reasons that I had Jake introduce the fact that I identify as a cisgender gay man is because I do a lot of advocacy for underrepresented students, especially queer students, and basically talking about the ways that they experience academia because there is a lot of inequity built into the infrastructure of higher ed. And I've spent a lot of time professionally working on basically changing the way that the system is designed so that we can be more inclusive and lead to a greater diversity of scientists in STEM, which is ultimately the driver of creative ideas, as well as creative and innovative collaboration, which I think you can all appreciate as sort of the bailiwick of the 21st century. So before I'll start talking about the research, I just want to make sure that I give credit to the various funding bodies, large and small, that have supported the research that I'll talk just a little bit about today. I'm mostly going to talk about natural history more than I will research per se. But as you can appreciate from these logos, there's been a diversity of vested interests in the work that I'll talk about today. And most recently, my work has been primarily supported by U.S. Department of Agriculture, specifically the National Wildlife Research Center, as well as USGS, so U.S. Geological Survey. Those two entities do a lot of focused work on invasive species. So at the end of my talk, I'll talk about some invasive snake work that we've been doing recently. And then most recently, it's been supported by a very large grant from the National Park Service. So I'll start off as the teacher that I am by asking you to think about what you see on the screen. So you'll notice that these are all vertebrates, but I want you to think about what they have in common in terms of what they're doing. What are these snapshots really showing you as imagery, right? So we would normally have like a live discussion about this and people would volunteer answers and all of that, but I hope that, that you've picked up on the fact that these animals are communicating. And more importantly, they're communicating within their species. So what they're doing is they're using sexual signals, these components of communication that function to reveal something about the signaler that reflects their quality as a potential mate. So one of my favorite examples that's on this slide is this singing mouse. Mice use vocalizations during courtship. So lots of different species of mice will do this. And the vocalizations reflect the quality of the singer. right? The same is true for many bird species, if not all bird species that use vocalizations during part of their courtship. And the most spectacular example, I think, comes from birds like the lyrebird, which have these remarkable integrated and multi-component sexual signal displays. My career has really been focused on two images that you might not have centered on immediately, this ghost knifefish that you see here, and then these newts, both of which use cryptic signals that we have to use very fine tools to appreciate. Specifically, in the ghost knifefish's case, they use electric waveforms that you have to detect with special equipment, and then these newts that you're watching here in a courtship display where there's a male and a female are using chemical signals. So my whole research sort of focus is really on the chemical signals that animals use to communicate through sexual signaling. Now, if you focus on this slide, you'll notice again, there are multiple vertebrate species on the slide, and I want you to think about what they all have in common and what they're doing. These are all vertebrates that are communicating using chemical signals. So for example, we know that beavers have a very distinct sexual signal that they use in their musk that they produce that reflects male quality. And then sort of a classic example comes from elephants that go into must whenever the males become reproductive, and they start to secrete these sex pheromones from this gland that indicate aspects of male quality to potential mates. But then there is another underlying connection between the species that are boxed in red here. And if any of you know anything about lampreys, that's a major clue in terms of what these species have in common. And what they have in common is that they are all representative invasive or pest species. So again, I mentioned my support, my financial support through USDA. I've been funded by them primarily to study invasive species chemical communication. So that's sort of the clue there. Now, this ultimately brings me to my favorite topic, which is sexual dimorphism. So the best example of sexual dimorphism that I love to talk to students about is human faces, right? So we know that Tyra Banks, for example, is a person with very characteristic feminized features. She has a high forehead, she has wide set eyes, and she has a narrow chin. These are all characteristics that we recognize as more feminine than masculine. But what you might not have known before this talk is that those features of the human face are actually sculpted during development by estrogens. So female typical sex hormones. Whenever those are high in circulation, the baby is typically born with this more feminized face. Contrast Zach Galifianakis, my favorite comedian ever. Zach represents these sort of classic androgenized or masculinized features, such as the prominent brow, excessive facial hair. These are androgenized features. These are features that are sculpted during development, but by testosterone and other androgens. So what I want you to appreciate here is the signals that animals display to potential mates are undergirded by sex hormones like testosterone and estrogen. Garter snakes, it turns out, are no different. They show strong size dimorphism. So here's a female garter snake that you see here. And here's the male garter snake who's starting to court her and trying to mate with her. Today, I'm going to unwrap a lot of the secrets that we understand about how snakes communicate with one another and how they sexually combat one another to lead to differential outcomes in reproduction. So for those that are really curious, I love to talk about elephant seals too, because elephant seals show really strong size dimorphism that's male biased. We're all familiar with the phenotypic differences between lions, between males and females. And then one of the coolest examples that's really small but significant are seen in newts. So here we have a female newt's forelimb versus a male newt's forelimb during the breeding season. And what I want you to notice, and hopefully you can pick up on, is that the ends of the toes of the newt are more elaborate. They appear to be covered in sort of a callus. Turns out those are called toe pads, and they're used to grip the female during reproduction. So it's pretty cool. So we can see lots of examples of these dimorphisms that enable us to tell which sex is more likely playing a direct role in reproduction rather than the passive entity. I'll also talk a lot about bird plumage later. So for those that are really curious about what my lab specializes in, I'm just going to go briefly over this slide. I work in 3 different areas of biology. I study animal behavior, I study pheromone chemistry, which for a lot of people is really boring, and I won't spend a lot of time on that today. And similarly, a lot of people get bored when I start talking about testosterone versus estrogens. But what I want you to understand is that these areas overlap in what I study. So we're interested in gene expression, but today I'm going to talk a lot about chemical ecology, or the interface between pheromones and behavior. And then I'll also talk a lot about courtship and mating, which is driven by sex hormones that command behavior to be acted, and then things that we can observe by studying animal behavior. So my research takes sort of trajectories through these disciplines to thread these disparate entities within biology together to create a compelling narrative. So we do lots of different things to study. So this is a technique sort of display. And what I want you to appreciate is that, is that we use a lot of fancy chemistry to understand the pheromones, but then today I'm going to spend a lot of time talking about bioassays, because as the title of this sort of seminar series indicates, you really care about the field. So I'll talk a lot about what we do in the field to understand sex behavior in the snakes. Welcome to my study system. This is a U-Haul design that the province of Manitoba decided to invest in as their premier advertisement for Come to Manitoba, right? This is how they wanted to compel people to increase tourism to Manitoba. Snakes. But what I want you to appreciate from this is that within Canada, Manitoba is really well known for these large concentrations that are not seen anywhere else in the world of garter snakes, right? And specifically, this is the largest sort of aggregate breeding for any vertebrate that we see on the planet. So I want you to just appreciate that you're looking at something at a massive scale on land that isn't seen anywhere else in the world. So that's what makes this system so spectacular. But my little claim to fame that I love to talk about is that this is actually a snake that was one of my study snakes when I was a PhD student. They contacted my PhD advisor, said, hey, do you have a snake? U-Haul needs to draw one, and we need a live model to study to be able to accurately depict what these snakes look like. So he said, Parker, ship him a snake. And so I put 474, that's her number. I packed her up in a box. I shipped her to U-Haul. And the funniest thing was when she got to U-Haul, they freaked out and they said, okay, she's alive. I said, good, she's a study animal. I need her back. And then they said, but we don't know what to do to take care of her. We don't know how to take care of her. I said, oh, that's easy. She eats fish and worms. And so I said, oh, great. How often do we have to feed her? I said, once a week, it'll be fine. They said, that's not enough. We're going to feed her more than that. So suffice to say, 474 lived the high life and then came back and weighed something like 20% more than she did when she left. So she had a really nice life as far as a garter snake's life is concerned. And now she's immortalized on the side of a U-Haul. So you might ask yourself, what is it like to be a garter snake for most of the year? I mean, I'm sure that's a question that burns all of you at night, right? I wonder what it's like to be a garter snake. Well, for most of the year, your life is going to look like this. You're going to be in the dark, and it's going to be between 2 and 4 degrees Celsius and at about 90% humidity because you're going to be underground. Manitoba, as you can imagine, has very intolerable conditions for most of the year because of its very high latitude compared to other places on the planet. So these garter snakes that are in the northernmost region of North America for reptiles, for reptile distributions, these snakes have to spend most of their life underground. And so the majority of their life will be spent like this in very cold conditions. So to be able to explosively breed and show up in the tens of thousands and start courting other animals and starting to mate in the spring, these snakes have to know when to come out of the ground. And the way that they know that is by the change in temperature that occurs in the ground. As spring slowly approaches in Manitoba, the ground starts to thaw. And as it does, temperatures slowly increase, and the snakes are tightly tuned to that change in temperature. And once the temperature starts to warm up, that's their cue to spawn emergence, right? So the snakes then emerge and they engage in robust reproductive behavior. And it looks like this. So every snake that you see in this image is a male. These are all of the snakes that have just come out of the ground and they're zooming around looking for other snakes, but specifically, they're looking for females. The question that should really pin in your mind immediately is, well, how are they finding females compared to all these other snakes that are present, right? Not to mention, there's a lot of male-male competition, right? This is the stuff that natural selection loves to work with, especially sexual selection. So we can study male behavior to understand a lot about female quality. So whenever we talk about sexual signals, that's the stuff that we really study. So all of these males have emerged from hibernation and they're looking for mating opportunities in the vicinity. They're looking for females. So again, in this area where we study the snakes, this is the entire distribution of the common garter snake in North and Central America. What I want you to appreciate is this is the widest distribution for a reptile in the Western Hemisphere. So there is no other species of reptile that has a greater distribution. We study the snakes here in this location, in the interlake region of Manitoba. And as you can imagine, it's very inhospitable for most of the year. But what makes this system so cool for someone like me who teaches undergraduates is that I can take students up here and we can study these snakes in their natural habitat at the den where they're coming out of the ground. So from a field biology perspective, it's a gold mine. There are no limits to what you can do. The only thing that limits you is the weather, which is actually kind of a challenge in Manitoba. But in general, we get nothing but clear sunny days with ambient temperatures, you know, around 5 to 10 degrees Celsius, and the snakes are just going at it. And I'll talk extensively about that now. So this is our field site. Very unglamorous. It's all limestone karst. And what I want you to appreciate here is that it looks like limestone rubble. Now I want you to take a second and try to count how many snakes you can see on the screen. So give it a good, I'll give you a good like 10, 15 seconds. Tell me how many snakes you can see. All right, so you either said 3 or 4, depending on if you counted this dead snake up here. Well, that's pretty easy, right? And this is unremarkable. But to see that many garter snakes in one spot would be pretty rare, I would say, in most places in the Western Hemisphere, right? But this is not what I'm talking about when I say mating aggregations. This is what I'm talking about. So here's an image taken from the same position, but a few days later, after the ground had sufficiently warmed up and the males had emerged. And what you can't see in this image is that snakes are all zooming around, and what they're doing is they're looking for females who are emerging from this limestone sinkhole, right? We really work on a rock quarry, but at the edge of this rock here is a large sinkhole, and there's another sinkhole right here, right off the shot. So these snakes are coming out of these sinkholes within the quarry, and they are looking for mates. So again, this is happening about May every year in the interlake region of Manitoba. And here is a female who's finally emerged. So what I want you to notice is she has so many males on her that you can barely see the female. So here she is, and these are all of the males who have found her and are now starting to court her, right? Including one male who's obviously missed out for the time being, right? And so these garter snakes essentially live 2 lives every year. So their annual cycle looks like this. In the spring, they spend about 4 to 6 weeks mating through these mating aggregations and lots of male-male competition. And then they will disperse to go forage. And they disperse 1 to 10 kilometers to feeding ponds and other large bodies of fresh water in the area, where they'll feed on fish and amphibians as well as earthworms in the vicinity, and then stock up as much energy as they can over those 3 to 4 months, because then they have to migrate back to the den and then overwinter. So most of their annual cycle, about 8 months of every year, is spent underground at 4 degrees Celsius in the dark, right? So they have a very limited active window in which to mate, forage, migrate, have babies if they're a female, and then migrate back, right? It's a really, really restricted time budget for a vertebrate. So we think of the snakes also as living 2 different lives. During the mating season, they're spending their time socially, undergoing courtship, looking for mates, and then they disperse and live solitary lives where they're largely asocial. So these snakes do not stick together. There are no familial sort of bonds. They don't show any sort of genetic identity-based or you know, relatedness-based aggregation, so they aren't social. And then they have to migrate back after this asocial period of their life every year. So it's a really fascinating life cycle just in and of itself. My lab studies the chemical changes that happen in the pheromone mixtures that are present for females and how males perceive them. But then we also do a lot of manipulation. So my lab specializes in changing the hormones that are within males versus females to see if we can alter this chemical communication. Currently, we're also doing some projects where we're looking at whether or not there are changes in the microbiome of the garter snakes between this social phase and then the asocial phase. And I don't have time to talk about that today, but for now, I'll just sort of tease you with that sort of question. So how are the microbes playing a role in the natural history cycle of the snake? The principal reason that we love to study the snakes in Manitoba is because of sample size. So in the spring, we can see hundreds of males aggregated near the den, and that's what you see here. This is a vintage clip of Bob Mason that I got off of YouTube, where Bob has gone to this mating or this basking pile of males, and he's picked them up to show just how dense these mating aggregations can be. And this is only one cluster of snakes that are in the vicinity of the den, right? So also, I want to point out that every snake that Bob is interacting with is a male. So there's extreme male bias at the den during the breeding season, but the only reason for that is because males linger at the den. Females emerge singly, they mate, and then they disperse. So I mentioned the breeding season is about 4 to 6 weeks long. That's based on how long males stay at the den looking for mating opportunities. Okay, so because of this high male-male competition, we get to see really cool stuff like this. So here you'll notice this is a female garter. By the way, I'm not monitoring the chat, and so if anybody has questions, you can just interrupt me, and I'll answer your question like while I'm talking. But I can't see the chat while I'm doing the presentation, but feel free to interrupt me and ask questions. So what you'll see here is this is a...
We had one question.
Yeah, I'm ready.
Jake asked if Bob's hands would be stinky after he...
Absolutely, yes, absolutely. Garter snakes are, they produce very specific defensive musk. And turns out that the musk is actually loaded with lots of different compounds that give specific odors to the musk. That's really cool stuff I've read recently. And the other thing is that depending on what they're eating, their musk will smell different. So it's actually based on their diet as well. Whenever we look at other snakes besides garter snakes, one of the worst smelling snakes that musk is water snakes. Water snakes eat tons of fish. And so because of that, and y'all have lots of water snakes in Maine. So, so water snakes will musk, and when they do, first of all, they're really good at flinging it at you. They can actually hit you in the face with it. They've got really good aim. Same thing is true for cottonmouths. If you've ever heard of a cottonmouth, these venomous snakes in the southern U.S. Cottonmouths will also fling it into your mouth. It's remarkable. Garter snakes don't have good aim at all. They just rub it all over you. But yeah, so snakes have that really powerful defensive musk that they'll release. And by the time these snakes come out of hibernation, that stuff has incubated for a long period of time and can be really, really potent whenever it gets all over you, for sure, yeah. But I also want to emphasize that the musk is not a pheromone. That's a really important distinction when we think about chemical signals. The musk is a defensive compound that's used in an interspecific way rather than intraspecific. Okay, so this is an example of what we call a mating ball of the garter snake. So here's a female surrounded by about 9 males. I think I counted once, it's like 9 or something, because you can see just a few heads poking in and out there. And so this is a single female being courted by multiple males. Automatically, male-male competition is very intense here, right? Lots of males looking for singly emerging females. That sets up a perfect scenario for sexual conflict, where the female will be under selective pressure to avoid males, whereas males need to be spending a lot of time trying to convince the female to copulate. That's what we think of in one example of what sexual conflict can be. Another way that sexual conflict in the system can arise is whenever we think about what happens after a male mates with the female. So here's a female garter snake's tail, and here's her cloaca right here. And inside that cloaca is a giant gelatinous plug. And this is what we call the mating plug. Whenever a male gets the opportunity to mate with a female, not only does he deposit sperm in her, he will also deposit a plug inside her cloaca that prevents her from being able to mate with other males. So that's a really great example of how the males have evolved a mechanism to counteract the fact that the female might mate with multiple males since she isn't male limited, right? Pretty cool. And if you're really astute, you notice that there's actually two little plugs here. There's one plug here and another plug here. This is a female who actually got doubly mated. Two males happened to penetrate her at the same time, and they both deposited their own copulatory plugs. This is extremely rare in the system, but it's a great example of how the males have evolved a mechanism to do potential mate guarding of a female to protect their sperm investment in that female's reproductive outcome. Okay, in addition to blocking females from being able to mate again, the mate order will also affect how many offspring are fertilized by a given male. So there's a mate order preference as well. Lastly, and most complicated, is that females can use stored sperm. So just because this female this year mated with these two males, it doesn't mean that she can't use sperm that were already in her reproductive tract to fertilize her eggs that year. So the sexual conflict that's occurring in this system is remarkable and manifold. There are multiple layers here. Another really cool thing that sort of weakens the effect of the copulatory plug is that it will dissolve over the course of 24 to 48 hours, and the female can then mate again. So if she lingers at the den while she waits for this plug to fall out or be dissolved, she can then follow up with additional matings, which is really cool. So there's lots of examples of how the sexual conflict has arisen and how these different mechanisms have evolved in the snakes. Now, another example of the sexual conflict that exists in the system. This is from my colleague, Chris Friesen. This is his bailiwick. Everything that I'm talking about sexual conflict, you should read his papers if you think this is really interesting. I'm going to now go into depth about how we assess these kinds of questions in this garter snake system. So here what you notice are the two intermittent hemipenes that a male has for copulation. You didn't think you were going to see snake penises, and I'm glad to show you something you weren't expecting. Okay, so you're welcome. You're getting your money's worth. Actually, it's free, so you're getting something for free. What I want you to notice is that, first of all, it doesn't really look like what we think of a phallus when it comes to vertebrates, and it's also covered with tiny hooks. So everything that you see protruding off the surface of this hemipene, and they're called a hemipene because they're actually divided in half, so they're halved, it's a halved penis, are hooks. So these are minor, tiny hooks that the male can use to anchor his hemipene inside the female. So this automatically shows that there is a degree of sexual conflict in this system because males have to anchor themselves inside the female reproductive tract in order for him to transfer his sperm, rather than just intromit and then transfer the sperm. He has to remain anchored in her, right? At the base of the hemipene is a large central spine that the male uses primarily for his purchase. So these smaller spines do provide additional anchoring, but this is the major player in that physical interaction between the hemipene and the female vaginal cavity. So now I'm going to talk about what Chris found in this study where they manipulated the presence or absence of this spine. This is some of the really cool field biology that we do in the garter snake system. So I'm trying to convince you that this is something you should want to study. This is something you should want to go and see. So what Chris did is he actually manipulated males by cutting off that spine. So he had groups of males that had the spine and groups of males that had the spine removed. And what I want you to notice is that when males don't have their spine, that large basal spine that you see here, the size of the copulatory plug they can make is reduced. So the spine enables the males to make this larger plug, which is really cool. But what I want you to notice is he only tested this in saline-treated females. Okay, so you should already be questioning, well, there must be another group that's going to come up on the slide, right? If you're really astute, you realize that. Before I do that, though, I want to point out that I'm talking about copulatory plug size from Chris's study, but really this was standardized to the length of the female, or to the length of the male, because larger males, meaning longer, produce larger copulatory plugs. So these things are associated with male dimensions of size. So you have to account for those. So Chris did. These studies are always tightly controlled. So I just want to point that out to you. So he tested females that were saline injected. He also gave females anesthesia in their cloaca to make sure that the female couldn't detect that she was being mated. And what I want you to notice is that whenever the male's spine is removed and the female is anesthetized, he can make an extremely large copulatory plug. So what this means is that whenever the spine is gone, but the female is anesthetized, the copulatory plug gets larger. Why? Because the female cannot fight the production of that copulatory plug. So this, again, highlights additional sexual conflict that's occurring in the system. I really wanted to talk about Chris's work today because I think it's remarkable. This is some of the coolest stuff that's ever come out of the system that, again, reveals some of the secrets about how snakes have sex and how they evolve to have better sex. That's really what I should have called this talk. Okay. So again, males have spines that enable them to make a large copulatory plug, and also the female can fight copulation. So she can sense when the copulatory plug is being produced, and she will fight that as long as she's not anesthetized, right? So it's really interesting. In addition to that, we can also study other aspects of the female's biology rather than the male's biology. So in a different study that Chris conducted in 2014, he treated females with Marcaine. Again, that's that anesthetic agent that can numb the female's cloaca. And he just looked at male copulation duration. So how long were the males in copula with the female? If she's anesthetized in her cloaca only, copulation persists for a longer period of time. This should clearly indicate to you that females are fighting copulation in this system. They are trying to avoid a long duration copulation. And why would they do that? Because they've evolved to avoid that aspect of this process of reproduction, so the duration of copulation. They need to make males fight for longer duration copulation, which leads to more fit males, right? So the females are imposing selection on males, specifically in their competitive ability. So I mentioned that the Marcaine is a local anesthetic that's injected in the cloaca that prevents the female from detecting that she's being copulated with. Now, Chris also studied in a different experiment the relative sperm count within the female, depending on what the female does when she's being copulated with, right? So when she's receiving copulation, females can do what we call body rolls. So in this example that you're seeing here, the female is being courted by multiple males. If she is unreceptive, which will happen if she's been mated or if she's just not going to reproduce that year, she will actually spin around her vertical axis. So it looks like a little barrel roll that the female will do. That's the best way that a female can disable male courtship or male contact with her, right? So she can barrel roll to then get away from the male. And it turns out that whenever females don't roll, when they don't roll on their own, the amount of sperm deposited inside the female is higher, right? So this shows that female behavior can help avoid male copulation and courtship. Therefore, this is a mechanism by which females impose male selection, right? Body rolling or not. The sperm count that was from this study was not just total sperm count, it was relativized to copulation duration, because the longer a male copulates, obviously the more sperm he's going to deposit in the female. So you have to account for that whenever you standardize for sperm count. Okay, so this is just some of the cool stuff that I think is really neat in our system. Now, I want you to think about what it's like to be a snake, and a male specifically, as you're looking for mates in this system. I mentioned before, this is a really homogeneous landscape. You're surrounded by males, and you've got to find the female. The way that males can know who's a female and who's not is based on what's on the skin of the snake in front of you. Specifically, what are the chemicals, the pheromones that are being produced that enable you to identify and choose your mate. This is a male who's chosen poorly. This is a male who's actually courting a hammer that I had used after I'd been handling a bunch of females. So I had taken female pheromone on my own hands on accident, grabbed a hammer, did what I needed to do, and then dropped the hammer, turned around, and this male is courting that hammer intensively. He thought that he had landed the mother lode of females, right? Like, this is the mothership. I'm going to copulate. I'm going to have as many offspring as I can, right? He stayed in this position, courting the hammer, for about 3 hours in full-blown sun. His body was hot to the touch, and he was convinced that he had found this sexy female because of the pheromones that were on the hammer. That, to me, was the clearest indication that the female's sexual signal, her pheromone, is the dominant signal over anything else that a male can pay attention to in the garter snake system. So we know that there's chemical sexual dimorphism. So females produce lots of compounds that you see here in what we call a GC trace. So this is from a gas chromatogram. And each peak represents a different compound. So this is what females can produce in their skin. When we look at males, they produce the same compounds, but they produce very little of them. So there's a difference in concentration, and there's a difference in composition that's a lot harder to appreciate whenever you're looking at very small numbers of compounds for males compared to females. So the question becomes, what aspects of the pheromone are important in informing males about female quality? Well, to understand that, we have to talk about some definitions. I'll talk a little bit about hormones versus pheromones. So both of these are chemical messengers. So they both act as chemicals to signal something to something else, right? So they're chemical signals that move from one place to another. Hormones travel in the bloodstream or from one cell to another. So this stuff occurs within the body. Whenever you hear the word endocrinology, this is the study of hormones. And these are things that travel within a body and go from like one cell type to another, or they go from a tissue to a different cell, etc., to signal something within the body. So the most common sex hormones that you'll hear a lot about today are estradiol, or E2, which is a female estrogen, versus T, or testosterone, which is a male androgen. And what I want you to appreciate here is that these hormones move throughout the bloodstream and affect different tissues. They go to different clusters of cells and they command them to do different things. A pheromone is also a chemical messenger, but it travels in the environment and from one organism to another. So these are considered external signals. These are things that are occurring outside the body. So in this example, this testosterone in this guy has traveled through his bloodstream to a specific cluster of cells in the armpits, and it signaled those armpits to release a pheromone, right? Humans do produce compounds that have pheromone properties, and they do come from our different odor-producing glands within the armpits. So the point here is that the hormone is not a pheromone, it's inducing the release of pheromones. The garter snake sex pheromone is a blend of lots of different compounds. There are 18 different things that we call methylketones in this blend of compounds that we can see in a GC trace. This stuff doesn't matter. It doesn't matter that you understand what compound is what. What I want you to appreciate is that there's lots of them. There's lots of different kinds. Each number represents the molecular weight of a very specific compound. Whenever we look at the structure, this is what the actual chemicals look like. So we have a long chain of carbons with a ketone at the end versus a long chain of carbon with a double bond. This may seem like a really insignificant chemical difference between those two compounds, but this is the difference between being unattractive as a female versus being attractive. That single change in the conformation of the pheromone molecule itself will make a female more attractive versus less attractive. And we think of that as the ratio of unsaturated to saturated compounds. The more unsaturated a female's profile is, the more courtship she'll receive from males. So I mentioned before that sex hormones can control pheromone production in the snakes. And remember that we have a very stark difference between males and females. Females produce lots of pheromones, males produce very little when we think about garter snakes. So we know from lots of studies that my lab has done that this pheromone production is highly sensitive to steroid hormone manipulation. We know that estrogens turn on or activate pheromone production in females, and that testosterone inhibits this process. So testosterone can shut it down, estrogens can turn it on. So if we take males, and my lab really specializes in what I'm going to talk about now, we take male garter snakes, which produce very little of anything, and we can turn them into attractive animals using a variety of experimental approaches. First of all, we can give them estradiol implants. We can give them female hormones in a little capsule, and those hormones will make that male produce female pheromone and become attractive. We can take them into the field, we can put them down in the den, and they get swarmed by males because they're producing abundant female sex hormone, abundant female sex pheromone. See, even I mess it up, okay. In addition, and more perplexing, if we castrate males, those males lose their ability to produce circulating testosterone and therefore cannot inhibit their own pheromone production. So the absence of testosterone makes males attractive. Pretty interesting, right? The idea that we remove an inhibitor and then therefore allow them to make sex pheromone. So males can make their own sex pheromone. All they need is either the signal that turns it on or the removal of the signal that turns it off, right? So this is the Frankenstein type stuff that I do in this garter snake system. Moreover, whenever you take a castrate animal who is attractive in 1 year, and if you give him a T implant, so again, that little capsule of testosterone, he will no longer be attractive and he'll stop making sex pheromone. So really, these snakes produce a pheromone based on the hormones that are circulating in their bodies. And that's just the take home from this slide. This phenomenon of the relationship between circulating hormones and the signals that are expressed to the outside world that are sexual in nature is common across vertebrates. This is my favorite example of that. So here what I want you to notice are two different ruffs. These are birds that produce sexually dimorphic plumage during the breeding season. So this is a male ruff outside the breeding season, and this is that same male, but during the breeding season, produces this elaborate collar of feathers. If you castrate a male ruff, he can't do this, right? So his production of this signal is turned on by testosterone in this species, right? In the garter snakes, testosterone is inhibiting their production of pheromones. We can also study this in other species like electric fish. Whenever electric fish are given testosterone, in this case, a very potent version of testosterone called DHT, they will actually start to produce higher frequency, therefore higher energy electric waveforms that they use in communication. And then in a more sort of elaborate example, you can give lizards different amounts of testosterone and cause them to produce more stronger male typical signals versus weaker female typical signals, depending on what hormones you give them or deprive them of, which is pretty cool. So what I want you to appreciate here is these are universal truths in vertebrates, that hormones directly regulate these signals that we know they use in reproduction. And to sort of elaborate on this from an evolutionary standpoint, I want you to appreciate that estrogens are actually the most ancient drivers of sexual signals. So here you're looking at multiple different groups of birds with the more recently evolved groups at the very end of the phylogeny, and then mapped onto the phylogeny are specific traits that regulate the plumage that we know to be sexually dimorphic in that group. So what I want you to appreciate is that in passerine birds, such as songbirds, birds that migrate every year, that sing every spring, their sexually dimorphic plumage is actually not controlled by androgens, it's controlled by other hormones like luteinizing hormone. So they have evolved away from the control of their sexually dimorphic plumage based on sex hormones alone. So they're more evolved in that way. They've evolved away from that control mechanism. More basal groups of birds like turkeys, mallards, as well as ostriches, they all have sexually dimorphic plumage that is estrogen dependent. So what does that mean? It means that whenever you look at a male and a female ostrich, you have to remind yourself that the female is producing dull plumage because she has estrogens. So think about that. We're used to seeing bright, showy plumage in a male, and then we automatically assume, well, that must be turned on by testosterone. That's actually not true in basal birds. If you castrate a male ostrich, he still produces that male plumage if he's an adult. However, the female will produce her dull brown plumage only if she has adequate amounts of estrogen. If she lacks those estrogens, she can't produce her quote unquote dull female plumage. So the ancient characteristic here is estrogen controlling female expression of female traits. And the question becomes, well, how deep is this evolutionary relationship between estrogen and these signals? My research has shown that in garter snakes, it's at least as ancient as before birds and reptiles diverged from one another, which is fascinating. And so because of this ancient relationship, I think of sexual signals being united in terms of their origins evolutionarily. So snakes and birds have a lot more in common than you probably thought before I came and spoke to you today about all the research that we do in this system. And I want you to appreciate that whenever we look at showy plumage in a bird, for example, you have to know something about the evolutionary lineage of that specific bird species to know if those signals you're seeing are directly controlled by something like testosterone versus a female's plumage that might be controlled by estrogens. Now I'm going to switch and talk about how we study sex in snakes, okay? And so what I want you to appreciate is that a lot of snakes, and pretty much all snakes, pretty much do it the same way, right? Sex without legs is tough. And therefore, there are very few things you can do to make sure that you stay in copulo. Some groups of snakes do biting, where the male grasps the female by the neck and holds her in position, and this actually triggers receptivity on the part of the female so that she gapes her cloaca and allows intromission. Other snakes use body size dimorphism to control their mates, such as in cobras, where the males are much bigger than females and will dominate the female through size. And then we have other challenges, such as having sex in water, right? These are two sea snakes. The male is much smaller than the female, and he's actively courting her because the sex pheromone that she produces, we actually know what the sex pheromone is in sea snakes, is a lipid that stays tight next to her skin, and so the males can stay in contact with her even in water. It's remarkable stuff. My favorite example, though, are the fact that mating balls, so large aggregations of males with a single female, are evolutionarily ancient. Anacondas, which are some of the oldest species of snakes on the planet, still display these mating aggregations. So this is a commonality across snakes. If they have the opportunity, many males will gather around the most attractive female and vie for copulation with her. So there are some really interesting evolutionary truisms within snakes. We also love studying the garter snakes and snake reproduction because they love to do it regardless of if you're present or not. So what I want you to appreciate in this video is that here there are males in Manitoba, and they're looking for the female, and Bob is holding the female here, again, a vintage clip from YouTube, and the male is now actively courting her, regardless of the fact that Bob's holding onto this snake. That is an awesome thing to have at your disposal when it comes to a research model. My students can study reproduction without worrying about their presence disrupting what the snakes are doing, right? So snake reproductive behavior tends to be very, very robust, especially in natural systems when you go out and observe them in nature. We can also study snake sex by giving them chemical challenges. So we can produce female sex pheromone in a Y-shaped maze, and we can see if males, when they get exposed to this scent, if they can follow it accurately, right? We can use Y mazes to study all kinds of stuff, and I'm going to elaborate on that in just a second when I'm going to show you how we study this with Burmese pythons. Also, I've been doing lots of behavior work with snakes for a very long time, and so if you have any questions about how we test this, or why do you use this tool instead of this tool, I'm happy to talk about all of these different things that we do, because I've actually done a lot of innovation in different venues where a new species presented new problems that we had to address, and it's something we're constantly sort of working through. In Manitoba, we also do a lot of arena trials where we set up in arrays these behavioral tents. These are 1 meter cubes that are bound together, where we can put a set number of males into this arena, and then we can just watch how intensely that group of males courts that specific female. So let's imagine that this is a female who was given testosterone, for an example, right? Over a set period of time, we can just count how many males are courting that female in specific blocks. And it's a really great way to generate tons of data very quickly. Any of you who know anything about how people study behavior, you can appreciate that sample size is a huge issue. And so being able to set up things in arrays like this give us tons of data that allow us to see the truest signal for a given question, which I think is really awesome when you think about experimental design and the confines and constraints of doing this in the field. I'll talk about mating ball tests in a second, because this is a really potent way that we assess how attractive an experimental animal is in the hardest challenge that they have to meet, which is at the den around those rocks that I just showed you those images of, right? So we actually go into the den and we see how competitive a single experimental animal is relative to other animals in the vicinity. We can also do things like field counts. So here's Bob actually putting pheromone onto a paper towel in this down in the den, and this is exactly where I took that picture that you saw previously. And what he's doing is he's going to wait and then count how many males come and investigate this paper towel that has female pheromone laid on it. It's a really, really cool way to do a lot of bizarre experiments that lead to really cool questions, as well as some fascinating answers about how snakes do what they do. Here are my students doing that mating ball test that I mentioned. So this is Holly, and what Holly's doing, she's holding an experimental animal here. And then what she's doing is she's counting how many males start to court that experimental animal over time. And so she will be tallying this number in her head, and then she'll tell the data recorder in the field, they write it down. And then Kenzie is the person who actively removes the snakes that are courting this stimulus animal, and then she puts them into a hopper behind her so that we do this without replacement, so we don't count the same male multiple times. So you think about field research, and this is not really what a lot of people think of. You think of like being behind a blind and like using binoculars. No. Not with snake sex. You can just get right in there and do what you need to do because the snakes don't care that you're there. And another fun thing is that sometimes whenever the snakes get really, really hot, like during the middle of the day, as my students get hot, we all get really, really warm standing in the sun. They'll camp out under you, and so you have to watch where you're sitting to make sure that the snakes that you're not sitting on a snake, because they like to get in the shade that we provide for them because they're right next to the den. From these trials, we can look at, for instance, how many males are attracted per minute. So we can actually estimate how attractive an animal is. So we call this the competition test. That's what you're seeing depicted here. So Holly actually has 2 snakes in her hands. She has a female who's here, who's covered in a lot of males, and then she has a stimulus animal, or the focal animal here, who is stretched out. That snake has anchored his head against a rock, and what we do over the course of a minute is we see how many animals come off of this mating ball onto the stimulus animal. And what I want you to appreciate here is we can test this across multiple groups, and here we get statistical differences. This FAD group, whatever that means, is less attractive than wild females that we tested at the same time, right? You would expect wild females to be the most attractive animals in the den, and they always are. We can never make a female more attractive than a wild female who's just come out of the den. That's a truism, and we don't know why. It's one of the more perplexing questions. Females given estrogen, females fed that are really fat, females that didn't have babies the previous year. It doesn't matter. They will never be as attractive as a wild female who's coming out of the ground. There's just something about that natural incubation period that makes them more attractive. We can also allow the mating ball to accumulate on the stimulus animal over a given period of time without disrupting them. I mentioned that Kenzie will go in here and she'll remove a male as he starts courting that stimulus animal, and she's counting in her head as she does that. We can also just set a timer and let it go. And if we do that, we see a similar change in attractivity across these experimental groups that I mentioned, right? So these are 2 different assays that tell us different things about what the snakes are doing. Also, I mentioned before that we can give males estrogens and we can make them attractive. In my 2012 study that I mentioned before, we compared control males to estrogen-treated males, and we saw a very clear difference between how competitive those males are in attracting male courtship in this den scenario. So I just wanted to give you an example of how we actually study this stuff, trying to be mindful of time. The snakes also do this in the lab, and so what I want you to appreciate here, these are lab hibernated snakes that we then warmed up and then allowed them to interact with one another. And what I want you to appreciate is that this is the same type of courtship behavior that you can see at the den. These males are trying to copulate with that female. In the other tank next door, not a whole lot's happening. While in the other tank, this female is just as attractive as this female was. So I'm going to play that for you again. And if you'll notice, the female's actually got tape around her cloaca. We have to do that because if she mates, those males will stop courting her because she becomes unattractive. Also, the female will become unreceptive to courtship and she'll start barrel rolling. which I already mentioned before, is a mechanism by which they can avoid copulation and therefore force selection on males. Currently, we're studying how male enhancement can be enacted in the snakes. So what we've done is in this group, what you didn't know is those males were color-coded based on their number. And so their number reflects the treatment that they were in. And we're studying how competitive those males were in courting. And we studied how competitive those males are in producing sperm. So we actually do sperm counts on these animals as well. And we look at sperm morphology. So this is just another fascinating thing that we're starting to get involved in, where we're looking at the different levels in which a male can be competitive. Maybe a male who's bad at courtship produces tons of sperm. Maybe a male who doesn't make a lot of sperm makes better sperm, right? So there's lots of interesting aspects of male competitiveness that we're now starting to study. And so in the interest of time, I'm just going to go ahead and stop there and answer some questions. Thank you so much for tuning in today. And I just really appreciate the opportunity to talk to you all. I'm going to go ahead and put up my contact information so that if anybody wants to contact me, you can email me.
Thanks so much, Rocky.
Yeah.
We had a question from Sandra.
Totally.
Are snakes induced ovulators?
That's an awesome question, and no. Females will have offspring even if they don't mate in the spring. So the females a female who has been in the lab for a year, has never had exposure to a male, will have babies that year. So no, she's not an induced ovulator technically. However, whenever a female gets mated, her estradiol levels will spike. She goes through a post-mating estradiol surge. That is the only dose of estradiol she'll get the entire year. So she resembles an induced ovulator in that way, that mating causes her estrogen levels to spike, but she doesn't necessarily have to mate to ovulate and then become gravid. So these garter snakes give live birth. So they gestate and then they don't lay eggs, they give live birth. So great question.
Awesome. And Sandra also was comparing felines that have
Totally.
spines along.
Yeah. Oh, interesting. Yeah, I wasn't thinking about that. Totally. Yeah. Yeah. So cats have spined penises too. And I don't really know what people know about sexual conflict in felines, but that would be interesting to know. But we definitely in snakes, we definitely see that in some species that show these explosive breeding aggregations, like natricine snakes, they have very spined hemipenes. And then in other species that don't display these large mating aggregations, their hemipenes tend to be less spiny, so less sort of medieval looking. So yeah.
Interesting.
Yeah.
And we had a question from Jake. How many young will one female produce approximately?
That's dependent on a lot of factors, but primarily female length. So the longer a female is, the more fecund she is. She will have more babies. It scales with body size. And so a given garter snake, the largest clutch that I've seen born in the lab was somewhere around 70 babies. And the species we work with, the red-sided garter snake, tends to have lots of babies, most of which don't survive. So even whenever you feed them adequately, most of them die. That's only true for this population of snakes that we work with. Red-sided garter snakes from other areas tend to have fewer babies that are larger when they're born, and so they have higher survivability.
And then I just wanted to draw attention to the raise your hand feature. If anyone wanted to utilize that, now would be the time. So, and we don't have any other queued questions at the moment. But we'll just give folks a couple of minutes just to make sure nobody wants to raise their hand and ask you a question directly, Rocky. But while we wait, just thank you so much for the presentation, taking the time today to sit with us and teach us so much. I mean, I feel like this topic would just go on, and it could.
I know. I tend to just go off for a long time. And,
there's so many intricacies that could be elaborated on. So it's been such a pleasure. So we're just getting some praise. And Miranda, if we don't have anybody with raised hands, we are just at the 5 o'clock mark. So Rocky, your timing was perfect.
And also, if anybody wants to hear about the Burmese python stuff, I can talk about that very quickly. I can show you like what we're doing, but no need to go beyond the time.
I mean,
so.
I don't think that would be a problem. Miranda, was there a raised hand?
Yes.
Okay.
Oh, great. Okay, so if you click on them, you should give them the option to either speak or they can
So Miranda needs to do that, not me.
Yes.
Yes, Miranda will do that.
Got it.
All right. So, Mark, you are currently muted, so you'll have to unmute on your own computer.
I just wanted to ask a quick question of Rocky as to the implications. I'm retired National Park Service. I started my
awesome, great.
And boy, what a mess down there today with the Burmese python.
Oh,
if you
absolutely. So did you want to ask a question about what we're doing down there, or you just want me to just sort of like give you a little synopsis?
A synopsis would be great.
Awesome, yeah. So one of the things that we're doing down in the Everglades, I'll just show. So my lab is now specializing in breaking snakes. So we're doing this. We're using chemistry and behavior to decipher how Burmese pythons are finding their mates, and we're doing this through a lot of behavioral work where we're putting them in Y mazes and giving them challenges and seeing like, can males distinguish between a female scent trail and a male scent trail? Turns out they can, right? And then we're using estrogens to manipulate males in captivity. So these are wild caught snakes that are kept for a brief period of time to try to make them attractive to other males, potentially to lure those males out from the wild, right? So we're using these tools to improve detection and detection probability. And the reason that detection is such a huge problem in the Everglades is because you're working in very dynamic systems that are dry for part of the year and then wet for another part of the year, as you already know, right? And so, for instance, here's a snake that we stumbled upon, and I don't think anybody can immediately figure out where the snake is. But this intern at USGS was super stoked because this is the first time she had gotten eyes on the snake in over 9 months of radiotelemetry with the snake. So she could get to the location of where the snake was, but she couldn't necessarily directly see the snake. And so if you are very astute, you noticed that the snake is actually at the base of this tree. I don't know if anybody had caught that. But this is a snake, like I said, that she had been tracking for months and had never gotten to see. And this was the day she finally got to see. It was the day that I was actually out with them. So we're using a combination of radiotelemetry as well as hormone manipulation to hopefully make males attractive to other males so that we can lure unidentified males toward those radiotelemetred animals and then remove them. In addition to that, we're also doing some work out in the actual Everglades near Daniel Beard Center, for anybody that's been down there. And here we set up tents with Y mazes in them to ask these ecological questions. Can males trail females? Can males tell the difference between an estrogen manipulated male and a normal male, et cetera, to see if we can manipulate the chemical signals the snakes are using. So that's what we've been doing in collaboration with National Park Service. But as I mentioned, the landscape is very dynamic. And the habitats are dynamic and extremely diverse. So deploying these cues, these chemical cues, pheromone baits, for example, presents a whole nother bear of challenges that we have to think about. So that's just a little taste of the stuff that we've been doing with National Park Service. So hopefully that answers your question or gave you a little bit of a clue as to what we've been doing.
That's awesome, Rocky.
Okay.
Miranda, did we have any other raised hands?
We don't have any more raised hands.
Okay. Well, I mean, you know, Rocky, if you wanted to go on for another 5 or 10 minutes, if you had more subject matter to present, you most certainly could.
Yeah, I'm good. I think that I talked about the Everglades stuff. I wanted to make sure that y'all could see that. One of the things I do want everyone that's still tuned in to appreciate is that, you know, Burmese pythons are only one example of an invasive snake species, and there are many that are extremely problematic, such as the brown tree snake on Guam. This species was responsible for the extirpation of all the native bird species on Guam, caused, you know, a silent night to occur on the island that was a bird watcher paradise for so long, but that snake got accidentally transported to Guam, has created just essentially ecological disaster. We also have the same thing happening on lots of islands because of boa constrictors that have been illegally transported to islands and have no native predators and abundant food supply, and similar situation, you're in islands, right? So this is happening in the Caribbean. Boas in the Caribbean are a huge problem. Also, down in Florida, one of the major concerns we have is that Burmese pythons are going to get to these isolated islands that are nearby, such as in the Keys, and potentially extirpate native bird species. In the, I can't remember. Sorry, but in off the coast of Spain, I just can't remember the islands. Canary Islands, thank you. In the Canary Islands, king snakes had been released accidentally and have bred and are a huge problem on that island, that island system. And then also in California, within a large continent, we have issues of invasive species such as water snakes in ponds that have shorebirds, et cetera, as well as native amphibians that the snakes are eating, and threatened fish species. So snakes represent a group of potentially extremely problematic vertebrates that once they become invasive, eradicating them is a massive problem. So I just want to sort of make people appreciate the fact that while snakes are fascinating and super interesting, they also can present really important challenges ecologically that we have to think about. And that's the reason that we do all this applied work to try to solve this. So, but thank you all for paying attention and coming. I really appreciate the opportunity.
Rocky, thank you so much. And Miranda, thank you so much for filling in for Lander this week. You were an absolute pleasure to have.
Thank you. This has been so interesting. It's not every day that you see close-ups of snake penises at work. So
there you go. You're welcome. I do what I can.
All right, and to our viewers, thank you so much for tuning in. And there will be a plug for next week, and there will be a follow-up email with the recording attachment for this soon to come. So thank you all so much, and everyone have a great evening.
Cool. All right. Bye. Thank you.